Hello students, in our previous video we have studied about cell membrane, cell wall, the shape and the types of cell and about nucleus. In this video we are going to study about cell organelles. So let us start. Let us start from the mitochondria. Mitochondria is the double membrane organelle that contains the two membranes. One is the outer membrane and another one is the inner membrane. It is also termed as the powerhouse of the cell. The outer membrane of mitochondria is smooth and composed of equal amount of phospholipids and protein. It bears various proteins termed as porins. These are the integral proteins, and this membrane is freely permeable for the exchange of substances or nutrients. The inner membrane of mitochondria is more complex. It is folded into number of folds that are named as cristae. There are many folds in inner membrane, or inner membrane is modified to form the small finger-like structures termed as cristae. These folds helps to increase the surface area for production of ATP molecules, and this mitochondria is also the site of oxidation of food, where the energy is stored in the form of ATP. Now, after the inner and the outer membrane, we have a space in between these two membranes that is termed as intermembranal space. It is a space between these two membranes that has the same composition as that of the cytoplasm. There is a difference between the protein content of the outer membrane, inner membrane, and this space. Now comes to the matrix. The matrix is a substance that is inside the inner membrane, that is present inside the mitochondria. It contains proteins and enzymes. Now we have the enzymes for the synthesis of ATP molecule, and the mitochondria also contain its own DNA and ribosomes. That is why it is also termed as semi-autonomous organelle. Now comes to the functions. The most important function of mitochondria is to produce energy. It helps the cell to maintain the proper concentration of calcium ions, and it also helps in making various other parts, like helpful in building the certain parts of the blood, and helpful in producing the hormones like testosterone, estrogen, and the mitochondria also plays the role in apoptosis, that is programmed cell death. Now come to the plastids. Plastids are of various types. The colored plastids are known as chloroplast and chromoplast, and the colorless are leucoplast. So the chloroplast are the disc-shaped organelle that contains the chlorophyll. As you know that the green plants contain chlorophyll to make their food by the process of photosynthesis. Chromoplast are the colorful plastids. They impart color to the fruits and vegetables. The leucoplast are the colorless. They are found in roots, seeds, and underground stem. Now look at the structure of the chloroplast. This chloroplast contains, as like to the mitochondria, similarly it has double membrane. It is a double membranous structure, and it bears its own DNA. And the ribosomes present in both mitochondria and chloroplast are 70 as ribosomes. Now, this chloroplast bear thylakoid. Thylakoid are the flattened disc-like structure, and these thylakoids are arranged in the form of stacks to increase the surface volume ratio of the thylakoid membrane, and these stacks are termed as grana. There are two photosystems, and these photosystems contains pigments. Now we have the stroma, that is the matrix or the substance present inside that contain various enzymes. And now we have the lamella, the stroma lamellae, that connects the various thylakoids together, as you are able to see here in the diagram. Now we have the endosymbiotic theory. This theory just states that there are certain organelles which are endosymbiotic. That means which are within together, they have the living substances within their body. We have mitochondria and chloroplast are the two organelles. So the modern eukaryotic cells evolved from simple phototrophic cells that ingest bacteria and cyanobacteria, and the prey was not digested, so the physiological processes of the endosymbionts were used by the host. Now we have endoplasmic reticulum. Endo means inside, plasmic, cytoplasm, within cytoplasm. Reticulum means the network. So the organelle that is present in the form of network, when it bears the ribosomes, it is termed as RER, rough, then surface is rough, and when the surface is smooth, then it is smooth endoplasmic reticulum. Here we have the difference between the both, that the smooth endoplasmic reticulum, the surface is smooth and it is not covered with ribosomes, and the in the rough endoplasmic reticulum the surface is rough as it is covered with the ribosomes. So here you have the differences, and the functional difference is that the smooth endoplasmic reticulum helps to synthesize the lipids, while the rough endoplasmic reticulum helps to synthesize the proteins as it bears the ribosomes. Now we have the Golgi apparatus. The Golgi apparatus were discovered by this Italian biologist, that is Camillo Golgi. In the plant cell, the Golgi apparatus is termed as dictysome, but in case of human or other animal cell, it is termed as Golgi apparatus. It is located in the cytoplasm, just next to the endoplasmic reticulum. The structure bears the cisternae, vesicle and tubules. It is semicircular in shape. It bears two end, one end is cis, another end is trans, as you are able to see in this picture, in the structure, that one end is cis, the cisternae, the end near to the cisternae is cis, and another end, opposite end, is termed as trans, and the vacuoles released from this are helpful in formation of the lysosomes or the vesicles. The principal function of Golgi apparatus is the secretion, and as it carries the secretory granules, secondly it is helpful in packaging and transportation of materials like proteins and polysaccharides within the cell, and it is also helpful in the formation of the plasma membrane, cell wall and lysosomes. Now come to the lysosomes. Lysosomes are the small vesicles that bears hydrolytic enzymes, digestive enzymes. The various enzymes that are present in lysosomes are nucleases, protein digesting enzymes, carbohydrate digesting enzyme, lipid digesting enzymes. So there are various membranous sacs budded off from the Golgi apparatus to form the lysosomes. Another function of lysosome, it helps in digestion, as it helps to digest the entire cell or the torn out cell. There are three different kind of lysosomes: primary, secondary and residual bodies. The endomembrane system, those systems which include endoplasmic reticulum, Golgi apparatus, lysosome and vacuole are termed as endomembranous system. So these four cell organelles are the part of endomembranous system, as they are helpful in the production of various macromolecules including proteins and lipids. Now we have the ribosomes. The ribosomes are the organelle that is a single membrane organelle, and this organelle is made up of two subunits, one larger, one smaller. We have two kind of ribosomes. The prokaryotic ribosomes are 70S and the eukaryotic are 80S. The prokaryotic ribosomes are made up of two subunits, 50S and 30S, and the eukaryotic ribosomes are made up of 60S and 80S subunits, 60 and 40S subunits. So these two ribosomes are different from each other, as the prokaryotic ribosomes are present in prokaryotes, mitochondria and chloroplast. Now, what is the function of these ribosomes? The ribosomes are involved in the synthesis of proteins. Now we have the functions of ribosomes. The ribosomes plays very important role in translating the genetic message. This genetic message is carried by the mRNA from nucleus to the cytoplasm. So this